So we've been talking about clustering, and today my goal is to finish up the clustering section and move on to the let's say next section, which is on structural aspects of things. But we'll talk about that a little later. Last class we talked about two classes of clustering algorithms, which are common and I think sort of intuitively appealing. One were these agglomeration-based things, which sort of took individual, put everything in its own cluster, merged close enough clusters that are deemed nearby by whatever distance measure you want to have, and then kept doing the merging until you had a small enough number of things to be clusters to be interesting. There was k-means clustering, where we sort of said originally we're going to be k-means. We sort of start with random centers, assign everybody to the nearest center, readjust what the center definition is, and keep repeating until this converged. Okay? And these were sort of, I think, sort of the main, let's say classical examples of, I will say distance-based clustering methods. Any questions about that? Either of those methods? Yeah.
The connection with the clustering to what we've been doing previously is to find the similarities of.
Okay, so the question is, why are we talking about clustering? I interpret clustering as being sort of the mother of all problems on microarray data. Okay, we talked about microarray data, but just to remind everybody, where do we have microarray data? Boom. Here's a typical microarray data problem. We've got each column represents a gene. Okay? We would like to discover sets of genes that have similar properties to them. Okay? Perhaps they are co-regulated, meaning that they're turned on and turned off at similar times. Okay? And the natural way to do this kind of an experiment is we had, in this case, about 16 data points on each gene. When was it turned on and when was it turned off? Okay? We then wanted to cluster the genes based on similarity. And when you did that, certain logical groupings emerged. Okay? And that is why you cluster things. Okay? Because you have a lot of data out there. You don't immediately see what the organization of it is. Okay? Clustering is the way to discover an organization, sort of the groups that are there. Okay? And in a lot of different kinds of data, there's groups going to be there. Pick a random type of data. Let's take a look at it. Someone give me a data source from anything. Okay? What? Scores that we get in class. Okay? So if I were to take a look at students here, and I had a large number of students in my class, and I see what their grades are in homeworks and finals, and I say cluster them, undoubtedly groupings that will be interesting will come out. There will be, of course, the A students who do everything well. There will be the hardworking but not very smart students, okay, who did very well in their homeworks and bad on the exams. There will be the people who didn't, goofed off, okay, I mean they did nothing on their homeworks and their exams were, okay, okay? My guess is that students fall into these categories. That's from 20 years of experience tells me that's how students break down. If you were new to this business, if you took a bunch of student data and you clustered it, those are the kind of groupings that I think would emerge, right? And discovering this is an interesting thing. That's why you would cluster, okay? To discover, geez, why are there all these students here who, are, getting good grades but not, on tests and not doing homeworks? Oh, there are a lot of people who are smart but lazy, is what I'd learned, right? What would be another data set that type of data source that you might want to cluster? Somebody give me a data source of something, on something in the world. World temperature. Okay, well now, that's what world temperature, what does temperature mean? one thing that I imagine people probably do is temperature by itself, the temperature at one point in time for a city is one-dimensional data about it, okay? I suspect that there probably are, what you call it, if you took a time series of temperature, okay, maybe the average monthly temperature, okay, for each city. Now you've got a 12-dimensional data set, right? And I imagine if you cluster cities by this, you will discover certain things. You will, first of all, discover nearby things probably sit in the same cluster. Because you're right, we probably have similar weather than Manhattan. But you will probably discover other things that you cluster. Like you will discover that big cities probably all have very similar weather to it compared to the weather distribution in the world. If you're building a city, you probably want it to be in a comfortable place, right? So there's probably notions of comfortable places, okay, that emerge from this. And there's probably notions of horrible places to live, that people only live because there is some economic reason to, okay? And I imagine that if you clustered these things, you would see interesting patterns emerge. And that's what the clustering, you would discover, I suspect, that there are certain kinds of cities are located in certain kinds of places, okay? And that kind of discovery is why you do clustering, okay? Any questions? Yes?
Is there any proximity to this drawing as far as where the genes are activated? And I know you were talking about time, but is there any...
spatial information? Yeah. So it turns out that, well, by spatial, you may very well mean spatial on the genome, okay?
Well, actually, I was talking about spatial in
the cell.
Of the being that it's in, actually.
Well, okay. I mean, there's...
Would all these tests be going on with the same...
These tests, okay, so in this particular experiment, these tests were done in yeast, okay, which is a single-celled organism. And so to the resolution of this kind of study, basically what's going on in the cell, okay? Now, it would seem perfectly reasonable to try to look at expression in different tissues. So here's another classical reason why you might cluster. Suppose, let's say now, we measure for each of the genes in humans. Okay, here we have the rows are going to be the genes. The columns are going to be the genes. The rows might be different tissue samples. Maybe we'll talk about skin cells, brain cells, okay, liver cells. Okay, I'm going to guess something, a few hundred types of tissues in the human body, right? All know they come from stem cells, right? This is exciting now. You have one basic cell that makes everything else. Probably in understanding how stem cells work, it would be interesting to know how closely related are different tissues. This would seem like a perfectly reasonable question. Are skin cells, is the expression in skin cells more similar to what goes on in brain cells or what goes on in bone cells? Okay? Or blood cells or something like this. Does this make sense? A natural thing to do would be, can you cluster the tissues, okay, based on similarity of expression? And you would probably see certain groups emerging, that certain cells express similar genes, okay? And that now becomes an interesting question. Why is it? Perhaps when they differentiated, there were similar signals that differentiated it. Discovering this underlying order is why you do clustering, okay? And that is a big picture thing to do in almost any data set. Questions about that? Okay. Anybody else have any? Clustering is a great thing to do, yeah.
So when microarray data is clustered,
yeah,
do they usually use two or three different types of clustering and see if all the clusters look the same? Okay,
so the question is now, how when you're clustering microarray data, what algorithm do they use? Do they use multiple algorithms? Do they use... There's a question, first of all, what do they do, and a question of what they might do if they were capable, okay? We'll talk today about the main clustering program that is used for microarray data, okay? And one of the options in there is to select which distance criteria you might use, okay? And so I would say a reasonable explorer of this probably should use different distance criteria and see how they stack up. Okay? You're allowed to use different forms of, it's an agglomerative clustering program. So different criteria. Do you want single linkage? Do you want multiple linkage? Okay? This is the kind of thing that a worthy explorer of this would want to see how robust or not their clustering is under different distance functions. Okay? And think hard about what the right distance functions are. Okay? Any questions about that? We'll talk about that a little bit later. Any questions about why we do clustering? It seems like an important... You should take this away at the very... Clustering is an important thing to do. That much should be clear. The microarray data is a classic example of why you want to do it. Okay? But you want to do it in everything else. We'll talk about phylogeny after Thanksgiving. And that is basically trying to figure out how species descend. What were the natural, relationships that were... The distance between species presumably reflects the clusters in species, presumably reflects some kind of an evolutionary heritage, right? Birds are similar. Why are birds similar among themselves? Because they're all birds, right? And so figuring out what taxonomical relationships are, okay, is a clustering-like thing, okay? So there's no question this should be recognized as important. The microarray data is the classic example of this. But I assure you that any data set you want, okay, can usually be meaningfully clustered in an interesting way. Any questions? Okay? So that's why a lot of work goes into clustering algorithms. Any questions? Okay.
What's the A, B, C, D, E mark in the bottom?
What were these A, B, C, D, and E marked? First of all, I stole this picture from someplace. Okay. They're marked up according to interest for somebody. But what I am going to guess what they are marked up for is they are marked up by the guy that did the study and said, hey, this cluster is so interesting. I am going to give it an A and talk about it. And maybe I'm going to discover these are all genes that operate in a particular phase of the cell cycle. Okay? That's probably what this means. I mean, I know the cell cycle has different phases that people talk about. Usually give like the letter names for it. I don't think it's A through E. Okay? But basically what that is saying is that these genes are similar enough. Someone said, hey, this cluster A is particularly interesting. These are all the ones that are upregulated at the end of the cell cycle, right? That's what it looks like here, okay? And that's why I'm going to guess what those are. That was probably not annotated by a program. That was presumably annotated by a guy, using a graphics tool. Any questions? Okay, fair enough. So what is the clustering algorithm stuff? We talked about these agglomerative methods and the k-means methods. There's one other class of methods that are, I think, are quite good, okay, and specifically for categorical data, but in general for lots of things, okay, including microarray data, certain aspects of microarray data, would be ones where you want to do what they call graph theoretic clustering. The vision here is you have a vertex for each element, okay, a gene, except in the case of what we've been talking about with microarray elements, a student in the case of grades, okay? And then you put an edge between every pair of things that are similar enough to be interesting, okay? You have some notion, similar enough may be that the distance is small, okay? That you may have a specific ad hoc distance function you may find reflects interesting. But once you get this, you get a graph, possibly a weighted graph, okay? With edges between similar things, the weights would reflect how similar they are. And then the question is there a way to interpret these edges as clusters? If you did everything right, each clique cluster would form a clique, okay, a complete graph, okay, where every one in the class is close to every other one in the class, okay? And if you did a great job with your similarity metric, you might have a world where you have several components, each separated, each dense within themselves, okay, and each separated. then your problem reduces to nothing more than finding connected components. The bigger problem is there will probably also be spurious edges, things that are from separate edges that are separate components that are close. And there will probably be missing edges, okay? Certain pairs that are not, for whatever reason, not in principle part of the same cluster, but our distance function was not sensitive enough to recognize that they were similar enough to belong in the same cluster. The reason why they're in the same cluster is the weight of all the other evidence, right? It's something like, you could imagine a world where, you have somebody on one end of the spectrum, somebody on the other end of the spectrum. They're all members of the same group, but these guys are fringe. These guys are very similar to those. This guy is very similar to all of these. Even though this guy and this guy are not very similar, there is still evidence based on the graph structure that they probably belong in the same cluster. Okay? And so our goal now is, how do you interpret one of these similarity graphs, okay, in terms of clustering? Two questions about that. So this should smell like a graph theory problem, and there are lots of graph theoretic algorithms that arise in these kinds of things. Okay? In principle, the idea to try to take one of these similarity graphs and partition them into clusters is to recognize that if there are spurious edges that aren't there, typically you're going to have two real dense clusters separated, connected by a small number of edges that really shouldn't be there. And so the algorithmic problem is to find which edges can you delete to somehow disconnect the graph. Does everybody get that idea? Okay. So if we think about it, one class to reconstruct the clusters are cut-based algorithms. We would like to find, given a graph, a small number of edges with the property that if you cut it, it will disconnect the graph. Does that make sense? Okay, we have a similarity graph, a small number of edges to disconnect the graph seems like a reasonable criteria, correct? The other thing that is good news is that algorithmically, given a graph, finding the smallest number of edges to cut to disconnect the graph turns out to be an easy problem. Easy in the sense that there is an efficient algorithm. Not easy in the sense that there is an easily explained efficient algorithm. Okay? Basically, there's a connection between a problem called maximum flow and finding the minimum cut. Let's take this network here and interpret it as a network of pipes, right? If I wanted to, let's say, take a look at this point S and this point T, and I put a hose in here, okay, and tried to, turn on the tap so water is rushing into this thing. If each one of these edges is interpreted as a pipe of, let's say, a certain capacity, right, and I put a drain over here, there is a certain rate at which water will drain through this thing. Does that kind of make sense? Okay. The limitation in terms of how much water, how fast water can flow from here to there in the steady state, okay, is going to be limited by the bottleneck, the smallest number, in some sense, the limited capacity to cut this thing. Does this kind of make sense? That if I want to have water flow from here to here, having a lot of pipes and stuff in there is fine, but there is no way I can get more than 2 units of flow from here to there if there is a cut here of 2 pipes that would disconnect it. Does that make sense? Okay? So there is this formal connection between the maximum flow through 2 points and the minimum cut between here and there. It's basically the same quantity. And so given that there are fast algorithms to find the maximum flow, polynomial time algorithm to find the maximum flow, There is also an algorithm, that same algorithm will find the minimum cut size between 2 points. And so given this, it's a realistic question to now say, given a similarity graph, find the smallest number of edges to cut to disconnect it. Okay? Any questions about that? How many people are sort of with me? Okay? So the good news is we could find the minimum cut. Actually, I think I'm doing this, right? And basically using network flow, that kind of does that thing. The bad news is that if I'm given a graph and I want to find what is the maximum cut, the minimum cut to disconnect the graph, usually it's going to look something like this. Okay? Where you slice off one vertex, where what we're really looking for, think about what we're really looking for. We have here our pretty dense graph. We have here our pretty dense graph, right? There's going to be a certain number of edges between them, right? Which is a lot sparser, right? It could very well be the case that the degree of this vertex here is less than the size of that blue cut. Does that kind of make sense? If you think about it, if this vertex is of degree, if this cluster had n elements, and this cluster had m elements, right? The maximum in cluster degree here is going to be m minus 1, right? The maximum in degree cluster here is going to be n minus 1, right? The maximum spanning cluster number of edges is how much? How many edges might span these two clusters? N times m. Does everybody agree with that? Very, very easy for the minimum cut to just slice off a vertex, rather than look for this relatively sparse thing that's separating a lot of vertices. Does everybody get that? Yes?
When you say one vertex, you mean the source of the target?
Well, okay, so the source of the target, what I really mean is, okay, so when I talk about clustering, the right way to think about this is network flow goes from one place to another. That means it determines the minimum cut between this, separating this vertex and that other vertex. Running n network flows will find me the overall weakest point in the graph. Okay? And that overall weakest point is logically the cut that I'm probably interested in. Okay? So that's why I'm dropping my use of source and sink here. Okay? Intuitively, I should probably run this for all sources and sinks. Okay? Any questions? So what is a better problem? Network flow, minimum cut is good, but it would tend to slice off single small vertices. Much better is looking for a problem where it respects the fact that you want clusters to be large. Does that kind of make sense? Intuitively, we want the right clusters to be, separation to be, a separator that is small relative to the size of the clusters involved. Okay? And that's probably the interesting place to cut this graph. Okay? Any questions? If you buy that argument, then that gets you into, what we would call graph partitioning algorithms, where you want to not only find a good cut, but take into account the size of the components. And there are many different ways to formalize that. But the one that I think is easiest to think about would be to instead take your graph, instead of thinking of it as a similarity graph, replace every edge by a non-edge, and every non-edge by an edge. That would be what we call complementing the graph. Okay? In this case, what do we want? A separation, if we had two clusters, if this was a cluster in the similarity graph, it would be, both sides would be very strongly connected, right? if we complement them, what was very strongly connected is now has no edges, right? And the gap between them, which used to have few edges, now has a lot of edges. Does everybody see that? Now I claim, if I take the complement graph, and I now look for not the minimum cut, but the maximum cut, this will now get me closer to what I want. Why is that? I am now going to want to try to cut as many edges as possible, right? Cutting many edges means that to get many edges, I need to have a lot of vertices on each side. There's a quadratic number of possible edges, right? And the only way that I can get a quadratic number of edges cut is by having a lot of vertices on both sides. Does everybody agree with that? And, so I claim that finding the maximum cut has the property, the maximum cut in the complement will have the property that it will cut through a lot of places that are dissimilar in the original, okay, while wanting to have things on different sides. Any questions? Yes?
So in the first case, when you are cutting only one node,
Yeah.
because it has only few edges. So when you take the complement of that node will have most number of edges.
So now that would have a lot of edges. You're right. Okay.
And then we cut it, so wouldn't the same node be cut off in the complement as well?
Well, okay, but the answer is, suppose, let's say we have a vertex that had a low degree before, right? You're telling me, yes, now it's going to want to have a high degree. But the question is the resulting desirable max cut going to be to put this one vertex on one side and everybody else on the other side? Is that what it's going to want to do? It's going to say, hey, look, wow, I've got, this one had, it were n vertices before, this one had degree 2. Look at me, I can cut n minus 2 vertices, edges, right? That's a lot. But it almost certainly would be more if there was any vertex in here that had degree greater than 2. It would pay for me to move it over here too, right? I'd lose the one edge that I get from moving it, right? But I gain all the edges these guys cut. So the natural thing here is going to be to want to create clusters where there's a lot of guys on each side. Does that make sense? Okay. Any questions about that?
Basically, in the middle you get almost n to n cuts when you take the complement. At the place where there was a path earlier, you would almost get n into n.
Yes. So, right. So the point is that if I had a desire, if in fact I had two well-formed clusters that were both big, when I took the complement of it, almost every, something quadratic and the number of edges between them would get cut. It would make me want to cut, okay, so as to leave off a big thing. Does everybody get that? Okay. So this is a sign that maximum cut is a better formulation for this. Turns out not necessarily the ultimate formulation if you really want to do graph partitioning. So the good news is this is probably going to get us closer to what we mean. The bad news is that maximum cut is an NP-complete problem, along with any other kind of, most of these other graph partitioning. Once you start saying, I want both sides to be big, okay, or I want one side to be really dense, this starts to smell like an NP-complete problem, okay? And so the world of graph partitioning is a world of heuristics, okay, for these kinds of problems, okay? But this is sort of gives you a little bit of a flavor of the kinds of things that we should be doing. Any questions about that? Okay. So these graph theoretic clusterings are good, especially when your data set is not naturally represented by points, by features of numerical dimensions, but by qualitative data. Okay? Because it's often easier to take two things and decide what their distance is by an ad hoc measure, let's say, that really interpret all the categorical data in terms of a point in space so that you can use a Euclidean distance or something like that. Any questions? Okay, any questions? So clustering is a big algorithmic area. There are a lot of papers on this kind of thing. And, but these, I think, are the major algorithms for it. Any questions? One other related technique that is sometimes, I'm going to call it here clustering, but I think it's more a classification technique, okay, that people should be aware of, is what I would call nearest neighbor classification. Let's say we live in a world where we have data. Let's think of one of the classic microarray applications we talked about. We have a bunch of patients who had cancer, a bunch of patients who were healthy. And for each one of these, we took a microarray sample of them, right? Now we've got a certain number of healthy points and a certain number of, and each one of these is a microarray, let's say something in 6,000 dimensions, right? Suppose now we sample a new patient who we don't have a diagnosis on. How can we figure out what the diagnosis of this new patient is, given the kind of training data that we have here? Does everybody see that that's a natural problem here, right? You have labeled data, okay? You have high-dimensional labeled data. In our microarray data, what would we have? We would have maybe 30 to 50 microarray samples, each with 6,000 or 10,000 or 20,000 genes measured, okay? We want to build a diagnostic thing. So the new patient that comes along, you can tell what they have, okay? Any questions about that? Does everybody see that that's a natural problem? Okay? So how can you do this? What would be a simple way to do this? Okay, or a reasonable way to now try to classify what this poor guy has. What? You say HMM. HMM would probably not be what I would be thinking about. The reason is because these features, I mean, HMM, I interpret it as being an automata where there was strings, there was sort of a coherence between what the measurement we had at the ith character and the measurement we had at the I plus first character, right? If we're dealing with a microarray, you've got, here's one gene, here's another gene, right? The position, the relative position of the feature on the microarray has no meaning. Does that make sense? The same ordering might be in the training set. But I still picture a world, it's not the case that a small automata relative to the string is likely to capture what is interesting about this. Okay? So I don't think it's an HMM problem. How else might you do this kind of thing? Yeah?
Take the average for each cluster that you already defined for each dimensional data set and then find.
Okay, so one possibility is you take all the healthy people and you average their microarrays, let's say, every coordinate-wise. You find a centroid. That's really what we're talking about, right? You find a centroid of these guys, okay? And you ask me, am I nearer to this centroid or to that centroid? Does that make sense? Okay? That would be one way to do it, okay? What's good or bad about that? Let's any thinking about it, okay? Right. So one problem is the question of if the data is all very uniform, the centroid represents it well, right? But if we live in a world where, you know, here are the, the cancer patients, and here are the other patients, okay? The centroid probably doesn't capture it as well as individual things, right? And particularly there might be subtypes within this data. There may be two underlying types of cancer people, right? There could be two different types of healthy people, right? Like, let's say male and female. Maybe this is a difference in what your microarrays look like, okay? It would not be surprising if there were a couple of logical clusters within each one of the labeled sets, okay? And if so, taking a centroid of that doesn't really capture the structure of it. Does that make sense? So maybe we would extend your idea to cluster each side and then take the centroid of it, okay? Or in the looking case, where each element stays part of its own thing, we can talk about what we would call a nearest neighbor classifier. This is a nice, simple idea that is pretty good. You take each sample. In the case of microarray data, each sample is a point in space. If it was microarray data in 6,000 genes, each array is a point in 6,000 dimensional space, right? And each point has a label: cancer, cancer, healthy, 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 cancer, healthy, cancer, right? Now, you put your new point in space, and you ask yourself, which is your nearest neighbor, right? Which point is closest to you in this? If you have a distance function between these points, there is a natural notion of which point is closest, okay? And based on that, let's say it happens to be this one. We would decide that this point could take the label of its nearest point. We could decide that this person is healthy. Any questions about that? So nearest neighbor classification is a way of assigning a label to the unknown sample by picking its nearest point. Any questions about that? Okay? The good thing about it is that it's fast and simple and easy, right? So long as you have labeled data. Okay? The bad thing is, it's not completely robust. Okay? Let's imagine a world where, your neighborhood looked like this. All of these were cancer things. There was one healthy person in here, probably because they made a mistake and diagnosed him as healthy. He died 2 weeks after the data was, after the diagnosis was made, right? And now we decide where you are over here, right? If your nearest neighbor was deemed healthy, then of course you're now deemed healthy, right? So how can we make this more robust? K nearest neighbors. Does everybody agree that it may be a more robust thing to do would be, instead of just taking one nearest neighbor, take 3, 5, 10, some number, okay, and vote among the neighbors, right? Or weight them according to how far their distances are. Like maybe if you're 1 unit from me and my second nearest neighbor is 10 units from me, maybe I shouldn't weight those equally. I should give more vote to the really close one. So there are a variety of ways you can sort of do this kind of thing. But basically it should be clear that, that if you do this kind of thing, you can make the nearest neighbor thing more robust. Okay? Any questions about that? Okay? And so nearest neighbor classifiers are a good way to solve these sort of classification problems. Okay? Typically when you've got small numbers of samples, because the cost of iterating and making the classification, I've got to compare me to all the samples that are there. Right? Any questions about that? Okay? Any questions? Okay? So, so basically, these kind of classifiers are useful. Conceptually, they get you into something that people in geometry call Voronoi diagrams. Okay? If you look back actually at the previous slide, this is a picture of a Voronoi diagram. It is a way of carving up space, okay, around every point, so that for every cell, the points around the Voronoi cell are all the points that are closest to one of the input points, okay, than any of the other ones. Does everybody see this? If we look at this particular example, this red region has this point as its source. For every point in this region, its closest neighbor, okay, is the same one. Does everybody agree with that? So one way you can speed up the kind of search query of what is my nearest neighbor might be take your points, build this diagram to carve up the points into cells. And for every new sample, just find out what cell you're in. Okay? Once we know what cell you're in, then we can give it the proper label.
What are the points in the center and at the vertices? What are the differences?
So what's supposed to be here in this one, the question is, what is it? I think what the question really is, hold on. Uh-oh, trouble. Okay, hold on. Uh-oh, trouble. Sorry about this. Crash. No. Okay. I hope this is good. Good. Okay, so what is this diagram? The way to interpret this picture is that the black dots represent real data points. Okay? The black lines, okay, and the points on the lines correspond to added information we get, the geometric partitioning of space, okay, to reflect these cells. So if I had these three locations, let's draw it like this, and I wanted to carve up space among these things, right? This is the prototype, you know, man, woman, and Martian, okay? Somehow the right way to carve up space is going to be something like this, right? All the points that are closer to man than woman, if we found the perpendicular bisector between these 2 points, right? That gave a line. And everybody that lied on this side of the line was closer to man than woman, right? These set of perpendicular bisectors carve up space. And if you delete the ones that didn't really matter, the parts that didn't matter, you had some kind of a space partition like this. That's really what does matter, right? So Voronoi diagram is building these bisectors using the appropriate parts. And you get to carve up the area into space. Any questions about that? Yeah?
How long does it take to recover the point to find which cell to point to?
So the question is, how much time does it take to search one of these things? The answer is it depends upon the dimension. Actually, the question of these Voronoi diagrams are great things in low numbers of dimensions. They get harder and higher numbers of dimensions. But if you have 2 dimensions, it turns out that if you search for this properly, you could actually find the right cell in essentially logarithmic time. Okay? How might I convince you of that? One possibility is you could imagine, I'm not going to really convince you, but let's say I built something on top of this thing and said, is your point on the left or the right of this line, right? And if I do this, I could in principle throw out about half the cells, and the only place where there's any debate are the cells that are in the squishy boundary. Is that right? So the claim should be that something binary search-like, okay, with emphasis on like, okay, should be able to let you find the right cell in essentially a logarithmic number of queries. Okay? Any questions? So explicitly, these Voronoi diagrams are great things if your dimensionality is low. So if you have 2 dimensions, this is a great thing. If you have microarray data and you have 6,000 dimensions, this is a lousy thing. Okay? And so you would do it explicitly by comparing points. Okay? In a modest number of dimensions, there are some clever algorithms for finding nearest neighbors quickly. Okay? But it gets to be a harder, it gets to be quite a bit harder as the dimensions get up. Any questions about this? Yes?
Is it always the case that you have these different kinds of clustering methods, so you apply all of them and see which one comes?
So the question is, which is the right clustering method to apply? Okay? And so one possibility I say is diligently you say, oh, I should implement them all and try all of them. And that's probably not the right answer. Okay? Is the answer I should implement one and say that's right? That's probably not the right answer either. But how do you make a decision here? Okay, I claim that, I would say that, actually, maybe we actually can get to this point now. What are the decisions you should make when you're doing clustering? This is really the question now. What should I do when I have to cluster data? Okay? You should ask yourself questions. One question is, how many clusters should there be? Okay? Do you know something about what you're looking for? Okay? If you say, I have no idea, this thing came from Mars, means clustering may not be such a good thing. Okay? If on the other hand, darn it, there's healthy people and sick people. There's going to be 2 clusters here. Find it. Okay? That makes a difference. You should be thinking about how do you take your data and, somehow deal with these large multidimensional data sets. So, the question that tends to get glossed over, which I mentioned briefly about normalization. If we add a bunch of different features on something, how do you bring it down so one of the dimensions doesn't dominate your clustering or your distances? We talked a little bit about z-scores. Remember that? This question is, how do you take your data to the point where it's likely to cluster in an interesting way? And there's lots of questions about how you do dimension reduction and how you do normalization and things like that. And that's what you need to think a little bit about. There's going to be questions about how much noise do you have. If you recognize that your data is going to be, if you have a small number of elements that are very noisy, you're probably going to want to do a complete linkage clustering ahead of single linkage clustering. Okay? So these are issues you want to think about. In general, one thing I believe pretty strongly actually is that the algorithm you use actually makes much less difference than getting the right distance function and the right data normalization measure. Okay? So, the different algorithms have different advantages and disadvantages. It's not that they don't have their role. But I think that in general, people tend to botch up more getting the wrong distance function or doing the wrong data normalization before you actually try to do the clustering. Okay? But all of these things should be explored in when it comes to your trying to cluster data. Okay? Any questions? My hope is still that you will get the opportunity to do that on a third short assignment. Okay? But we'll see about that next class. I'm trying to figure out one. What?
Does that do next semester?
Anyway, the others would be short, but just to give you some experience, sort of twiddling different algorithms and seeing what happens. We will see if we can. We're trying to make it short. Okay? Any questions about that? Okay? It will be short. If I give it to you, it will be short. I promise you that much. Okay, any questions? So let's look at a case study of clustering microarray data. Okay? I'm curious as to what the, if you look at microarray data, Affymetrix is the company that makes those microarrays. Originally, they just made microarrays and they gave you images of them and they said, you go analyze them. But as time went by, everybody started writing their own clustering algorithm. Eventually, there emerged, reasonable sources of, tools for people to use. The most important early and still probably used algorithm for microarray clustering is something, it's actually a pair of programs called cluster and tree view, okay? That, when you see these pictures of these green and red microarray things, these nice, long, beautiful displays of microarrays in grid clustered where the rows are clustered, that's probably cluster. And basically, it's 2 different things. There is a cluster thing that does the clustering the way that we've talked about it. There is a separate component of actually visualizing these clusters that we should recognize is actually a different algorithmic problem than what we've actually been talking about so far. Okay? So how do these things work? Cluster, okay, if you look at cluster today, it gives you a suite of different algorithms, different distance measures. microarray data, what was sort of the recommended distance function was not really Euclidean distance, but was the correlation, okay, coefficient. So you had here, one cluster, you had here another cluster, or, gene or cluster. You had 6,000 dimensions or 10,000 dimensions. The claim that they used to measure a similarity or dissimilarity was the correlation coefficient, where they normalized the data by basically taking the log of the red-green ratio. So what does this mean? In microarray data, it's hard sometimes to measure exactly how much RNA things are sticking to. Why is that? We agreed that a microarray patch was sort of this set of DNA probes that were stuck to it, right? And that we were going to take a bit of solution and wash, we had our target RNA. We made this radioactive, right, or fluorescent. And we then poured the goop over this thing. Things would bind to these hairs, right? Note that if there was more RNA in the sample here than there was on the plate, all the hairs would get bound to and it would just light up. Does everybody agree with that? You'd quickly run out of resolution here, right? Suppose you've got a million probe molecules and 10 million RNA molecules in your target, right? Every probe is probably going to get lit up and you lose your resolution. Does that kind of make sense? Okay. Do people understand what I'm saying with that? So the way that they got around this is kind of interesting. What they would do is, instead of just taking one sample, okay, of the, RNA and making it, lighting it up, they would take the one that you were interested in and a reference target. Let's say that you had one person's sample who you knew was healthy, right? You have one, let's say one reference person, let's say Skiena's, okay? And you had your other sample that was unknown, right, that you were interested in. What you would do is take a sample of Skiena's RNA and radio label that red, and then take the target that the other one that the new array, and label that green. Then what would happen is, when you had your probes here, even if it got saturated, if you poured in an equal amount of Skiena's stuff and an equal amount of the other one, if this gene was expressed the same as in Skiena, as in your sample, you would expect that about half of these things would be green, and about half of them would be red. Does that make sense? And so you can now measure the ratio of how much expression is there relative to the reference, okay? And that gives you potentially more information if you choose your reference in a reasonable way, right? You want to know, in this sample, is this gene upregulated, meaning it's making more than it usually does, or downregulated, making less, okay? So the data that they usually assumed were basically red-green ratio data. One neat thing about a red-green ratio is it's something about x over y. When they're equal, the value is 1. When there's a lot more x, the value is big. When there's a lot more y, the value is small. If you take the logarithm of this thing, what's kind of neat is that if x is overexpressed, you get a positive value. If x is downregulated, you get a negative value. And if it's just normal, you get a 0. Does that kind of make sense? So the data normalization here was actually important. They took the logarithms of it, and that made a reasonable thing. Any questions about that? Okay? So that kind of transformation would make a difference. Note that if you didn't take the log of it, even if you just did the, note that there's a problem here. If you say, oh, I don't want to take the logarithm of it. What's the problem? an upregulated gene would have a value of 10, right? An unregulated gene would have a value of 0.1, right? Which is further from normal? Okay? Normal is 1, is that right? So then 10 looks like it's further from normal, but that's only an illusion, right? Because both of them are tenfold different than normal, right? Does everybody see that? So in order to make, let's say, not weird out your distance function by having the artificial notion that upregulated, you can get a bigger ratio than downregulated. That's why you needed to take the log, actually. Is that kind of clear? Okay. Normal was going to be 1. 10 times normal would be 10. 1/10 normal would be 0.1. Unless you take the log of these things, this looks like a significantly different thing than the other, when all it is really changing the difference between red and green. Okay? Any questions about that? So proper data normalization is important. They used, as I said, correlation coefficient to measure the similarity of these microarrays, okay, instead of something like distances. Now, I like distances because I'm a computer scientist and I think I want metrics and stuff. One thing that's interesting about microarray data is it's kind of a weird thing that each data point costs $500, right? But each would give you something on the order of 10,000 measurements, right? So if you think about it, the number of dimensions is much higher in general than the number of samples. If I wanted 10,000 samples, how much would it cost at $500 a pop? 5 million, right? And I don't get that kind of money. Very few people get that kind of money to do experiments with, right? So what's kind of weird about microarray data is that you have a small number of samples, but a large number of dimensions. That's usually not the way it is. I mean, think about something else. Think about students, right? I have a huge number of students, right? But I only get to give you 3 homeworks and two midterms, right? Okay? 2 homework. Okay. I get to give you 3, whether I will. But anyway, I will. Okay. But you get the point that usually the number of dimensions is smaller. We're used to thinking of a world where the number of dimensions is smaller than the number of samples, right? What else would be a data set? Was there any other data set you've ever seen? Stock market. Well, stock market data is now interesting. What's interesting about it is that also may have some of this component depending upon what the dimensions are, right? If the dimension is its price every day, then there's only S&P 500 features, right? Their prices over the last 100 years, that would be 100 times 365 days, right? You could have a world where the number of features is large relative to the number of dimensions, right? But probably you're better off refining the features into something more meaningful if you want to cluster or deal with it. Okay? So bottom line, correlations tend to be a good thing when your dimensions are higher than the number of points, would be my argument. Sort of a hand-waving argument based on that. Okay? I mean, with stock market, notice with time series, you're used to computing correlations of time series because time series tend to be long. The microarray data has the similar property that you have a large number of dimensions.
These points are the points on the microarray, each of them.
Right, so each dimension would be the red-green ratio for that particular patch, right? For that particular probe. That would be the value of each dimension. Okay? Any questions about that? Right, the number of sample points would be the number of well, okay, it depends what you're doing with this, how you're clustering. Let's think about this. In a microarray experiment, what you would typically have is each array measures a different gene. Is that right? So here you've got genes on this side. Here you've got patients or tissues, right? Patients or time, in the case of the cell cycle experiment, or tissue types, if we wanted to cluster different tissues, right? We would each one of these on these rows is going to correspond to an array. Each row is $500, right? But for your $500, you get 6,000 or 10,000, or depending upon what your array is, a certain number of thousands or tens of thousands of features, right? And so if you're clustering arrays by genes, you get a high-dimensional data set. If you're clustering genes by arrays, it's less clear it's a high-dimensional data set. Any questions? Okay. So what is it that Clustl did? What was the algorithm that they sort of preferred? This was, again, an example of the output of these kinds of things. Okay? You can see here that here you've got, what I'm going to guess is these are probably arrays. These are 6,000 genes, right? These are the clusters that were found. And note that they were displayed in a nice way. Okay? If you look at these things, these are the genes within a cluster. Okay? This is what the TreeView program would give you. Okay? Now, note that with any of our clustering algorithms, in this display there is a problem that you want to linearly order the genes within a cluster, right? So that it shows you something that's interesting. What is the right way to order the genes in the cluster? Okay? Does it make sense that this is still something we haven't really talked about? If we had been using k-means clustering, right? You could imagine a world where this might be one cluster, this might be another cluster, this might be another cluster. But how do you order the genes within a cluster for display? Is it clear that that's something that hasn't been covered yet? Okay? But is an important part of making these beautiful pictures. Any questions? Okay? So how do they do it? The clustering algorithm that they used in TreeView, okay, cluster, or at least in the as a default, they use an agglomerative clustering method. So it's from the individual elements merged up, okay? And what they used was basically something akin to, there are lots of different criteria for merging clusters. The one that they used was basically each cluster was going to be represented by something like its centroid, okay? Basically, its centroid. That was the average value of if I have a cluster, I've got a bunch of elements in here. I'm going to represent my cluster by its centroid. And then, if I want to merge two clusters, how do I merge the two things? Okay? If this was a centroid of a cluster with 10 genes, and this was a centroid of a cluster with 50 genes, okay, it should be clear I shouldn't just take the average of this and this, right? Because somehow what the meaningful of the centroid is, this represents a lot more genes in the merged cluster, right? So basically they would do something by merging the components, okay, weighting the merged components based on how many genes were in each one in recomputing the centroid. Any questions about that? Okay? So basically it's agglomerative clustering using something that I would call essentially the centroid measure. Any question? And there's certain weirdnesses for alphabet-specific things. One thing that happens in microarray data is sometimes the values are missing. Okay? Imagine a world where here is a microarray, and somebody spat on it before they actually ran it, right? All these cells would be ruined, okay? And you could imagine that there's a lot of, at certain points, sometimes certain data doesn't show up. And so you need special ways of dealing with missing data and things like that. Okay? But bottom line, that's sort of the pragmatics here that we would have. Any questions? Yes.
Should centroid be a point instead of a line?
So a centroid is a point in certain number of dimensions, right? So when you say something is a point, 10, 15, 3, 2, 1 is a point, right? It's an array of values. That's the way I would normally interpret what a point is. It's an array of values, right? And so in this model, what I'm picturing is each column is a dimension. The centroid is averaging all the values in that column, right? That's how I would get that interpretation. Any questions about it? Any other questions about this thing? There is this the amusing thing that I didn't talk about, which I'll just talk about briefly because it is amusing, is how they produce the pretty pictures. Okay? If it's an agglomerative clustering algorithm, that means that we're going to have a tree, right? There is a tree because at each step we merge certain genes together and certain clusters together. Is that right? So that's why when you saw with my original clustering, you saw that there was this tree associated with it, right? And so in drawing the matrix, it may make sense to think, well, gee, why don't I just draw the tree like it's a tree and have that specify what my genes are, the order of my genes. Does everybody see that? Okay? You see what I mean by this? If I have my tree I can draw in a nice non-crossing way, right? If I just use that to specify the order of things, my picture will sort of reflect the structure of my clustering. But there is a degree of freedom that I have in making the drawing that I haven't taken advantage of here. Note that when I'm drawing it, if I have this node here, it doesn't tell me which child is on the left and which child is on the right. Does it kind of make sense? If I want to do a drawing, okay, if this is gene 1, gene 2, gene 3, gene 4, what drawings are consistent with the tree, right? Putting gene 1 next to gene 3 is not consistent with it, right? Unless I reverse this node and reverse this node. Why couldn't I have gene 2, gene 1, gene 3, gene 4? Does everybody see that? So the freedom that I have to make my picture nice is I can do a reversal at each one of these nodes, what is left or what is right. Does everybody see that? And it turns out the key to making these drawings beautiful is figuring out what's the right way to reverse these nodes. Okay? What is the beautiful cost? A picture is beautiful is if this gene and this gene are very similar to each other, right? The picture is going to be beautiful if this looks a lot like that one, right? So my question, my algorithmic problem that you need to solve is basically how do you flip these things so as to come up with an ordering, a TSP-like ordering, where you have a low cost between neighboring things. Okay? Any questions about that? And the interesting thing is that if you do it right, okay, you can do this with dynamic programming and find the best order relatively efficiently using this dynamic program, which I don't think I'm going to go into. Any questions about that? Actually, maybe I will at this time. Okay? Any questions about that? Any questions about clustering or anything like this?
If C3 and C2 are similar genes, if G3 and G2 are similar genes, won't they be the assumption of the same one?
Well, you could imagine still that this is similar to this. Well, let's imagine, first of all, recognize that this is a small part of the picture, right? There were 6,000 genes in this thing, right? And so making it pretty means, in the course of agglomerating a meaningful cluster, right? All of these guys under here may be logically in a same cluster, right? But to see the coherence of the cluster, the two most distant members of the cluster probably still don't look quite as nice next to each other as there are intermediate ones, right? So the claim is that the right visualization involves a linear order of this, okay, where you have as little difference as you move along that, okay? Any questions about that? Any questions about clustering in general? Okay? Why it's good? Anything we've done in here? Okay? Tell you what, since I have a small number of minutes, let me explain the dynamic program rather than go into something else, okay? Just because dynamic programming is a good thing. How would we find the optimal tree? Okay? Given a tree, I'm given a tree, and I want to flip these nodes around, okay? So that I come up with the optimal linear order from left to right. Does everybody agree what my problem is? Now, finding the optimal order from left to right should smell to you like a traveling salesman problem. Why is that? What's the traveling salesman problem? That was where I had a bunch of cities I wanted to visit. I wanted to visit minimum total cost. Does everybody agree with that? I think I've talked about, I may have mentioned that in here. If not, did I mention that? Okay, does everybody know what I mean by traveling salesman problem? So if we think about it, we could imagine that the traveling salesman problem is every gene is going to be a vertex, okay? There is going to be an edge weight between two vertices based on how much they cost, right? And the right ordering for this matrix to be beautiful would be the one where the neighbor cost from neighbor to neighbor is as low as possible. Does everybody agree with that? So that would suggest that if I solve a traveling salesman problem, I solve my drawing problem, which is good, right? But traveling salesman is entity-complete, which is bad, right? But I have a tree here that I am trying to respect, right? So it's not any ordering that suffices. It's the ordering according to this tree. So what are we going to do? I'm going to define now a recurrence function, a function. Let's let C sub R ij be the cheapest way to order a subtree labeled R with a root of R, where gene I is on the left and gene J is on the right. Okay? Let's think about what I'm saying here in my function is. You give me your tree. For every node R, I'm going to find the best way to draw that subtree, where for every leaf below it, every pair of leaves I and J, I am going to figure out the cheapest cost of this subtree with I on the left and J on the right. Okay? That's my criteria. Any questions about that? If I had this function and knew how to compute it, what would be the best possible drawing for the entire tree? It would be what? It would be the min over all I and J, right, of the cost from the absolute root to ij, right? Does everybody see that? So if I can compute this thing, I can find the best overall drawing. And what is the cost of this thing? I claim that Cijr is going to be the min over all children possible K and L. So let's think about it. I'm given a node. If I want to go from the left to the right, I is going to have to be in the left child, and J is going to have to be in the right child. Does everybody agree with that? There's no way I can respect the tree. My left and right vertices were both on the same cluster. Does everybody agree with that? Okay. So what is it going to be? My claim is that I'm going to want to know the cost of this is going to be, of any drawing of this type, it's going to put I on the left, some vertex K from the left side over here. The other subtree will have J on the right, and some vertex L from the subtree that is going to be kissing the subtree vertex over there. Does that kind of make sense? The cost of what goes on in here is irrelevant to us. The total cost is going to depend upon what the outer vertices are. If so, the cost is going to be the cost of the edge connecting R, K, and L, and the cost of going from I to K on the left, and going